Okay, so for those of you who are just coming in, welcome to the Plant Sciences session, subsession number 2. And we're moving on to a talk by Sarah Swope of Mills College. And the title is Local Adaptation versus, Local Adaptation and Inbreeding Depression in the Threatened Tiburon Mariposa Lily, Caloportus tiburonensis.
Pull it down. There you go.
Is that better?
Yeah.
Okay, great, thanks. Okay, so I'm going to talk about some work that we've been doing with this plant for about 4 years now. As Ellen mentioned, it is the Tiburon mariposa lily, or Caloportus tiburonensis. It is restricted to serpentine soils, and it is endemic to a single site in Marin County. So first I want to talk a little bit about some of the questions that we've been exploring around its evolutionary ecology, and then I want to try and link that to some work that we've been doing on this population biology, and then talk about the conservation implications of this work. So one of the more fundamental questions in evolutionary ecology is, under what circumstances is gene flow, the movement of pollen or seeds from one population to another, good for a population or bad for a population? And I mentioned this plant is restricted to serpentine soils, as are a lot of the species of concern in California. And serpentine soils are, of course, very stressful places for plants. And abiotic stress such as this tends to have a strong influence on the evolution of plant populations, often driving local adaptation. So when you have strong local adaptation, gene flow between populations can actually be bad for populations because it can introduce maladapted alleles, and it can break up co-adapted gene complexes. On the other hand, serpentine soils tend to be very patchily distributed across the landscape, and so the plants adapted to those soils are also patchily distributed, or kind of naturally fragmented. So that presents a very different scenario. You often have these kind of small or isolated or naturally fragmented populations, and being small and isolated can cause a population to lose genetic diversity, which can then lead to inbreeding depression. And in this case, gene flow can actually be good for the populations because it can increase genetic variation by introducing novel genetic material. It can create novel genotypes, and it can alleviate inbreeding depression. So this is a plant that we're working with. I'm going to call it Caddy from here on out. It is a fairly long-lived perennial. Plants live to be about 10 or 12 years old. It flowers infrequently. It appears that the stress of flowering on these serpentine soils is so much that the plant typically goes dormant or is vegetative the year following flowering. It is primarily outcrossing, although it does do this kind of interesting thing of asexual reproduction. So like all lilies, it grows from a bulb, and that bulb, when it gets large enough, can split. And so what you end up with is two individual plants growing right next to one another, which demographically you can treat as individuals, independent individuals, but are genetically identical to each other. And we think that this becomes kind of an important piece of the story later on. Okay, so serpentine soils are infamous for having very low concentrations of essential nutrients, low water holding capacity, high pH, and high concentrations of toxic heavy metals. In fact, heavy metal concentration is oftentimes on par with what you'd find in mine tailings. So these are very stressful places for plants to live, and they have to evolve to tolerate those conditions. So for those of you who don't know, this plant is found only on Ring Mountain, which is right there. On the Tiburon Peninsula, we believe that it was only ever found on Ring Mountain. And because serpentine soils are patchily distributed across Ring Mountain, these populations, or each patch kind of supports its own semi-independent population, which range in size, we think, from maybe around 250 individuals up to several thousand individuals. So we're working across the full range of environmental conditions that this plant has to contend with. So this is an overview of Ring Mountain, and you can see it's surrounded by suburban development. I'm only going to introduce you to the 3 populations I'm going to be talking about today, although we've identified what we think is pretty much 7 largely independent populations. So a site that we call the nursery site, and another that we call the burn site. These are large populations with very high densities of plants. And then I'm going to mention this other population, what we call the Taylor population, which is much smaller, much lower plant density and considerably more isolated than the other populations. Okay, so plants that, restricted to these serpentine soils, but not all serpentine is created equal. There can be an awful lot of variation in serpentine soil chemistry from one patch to another. I'm going to show you some data to back that up. So these are results from our own chemical analyses, and I'm going to show you results from the nursery and the burn site. So this is a concentration of nickel, which is toxic to plants. And there's two things I want you to see. First, this red line here indicates nickel concentrations in typical non-serpentine soils. So I want you to see, first of all, that the concentrations of nickel at both of these sites is very, very high, but it's higher at the burn than at the nursery site. That was true, that general pattern of heavy metal concentration being higher at the burn than at the nursery was true for all 16 heavy metals that we looked at. I'm just showing you 4 semi-randomly chosen ones to illustrate that point. So not only did the burn site have higher concentrations of heavy metals, it also had lower concentrations of essential nutrients. So the way we're kind of thinking about it right now is that the burn might be a more stressful place for plants to live than the nursery site. Although it's all serpentine, so it's all pretty stressful. Okay, but we wanted to look at this soil chemical environment, kind of move beyond the simple ANOVA framework and look at it in a more comprehensive way. So we did an NMS ordination and we included concentrations of 25 essential nutrients and heavy metals. And for those of you who are unfamiliar with these ordinations, quite simply, the analysis will consider all 25 of these elements and we'll plot samples either really close to one another if they're chemically very similar to each other, or far apart from one another if they're chemically very different from one another. Okay, so let's look first at the burn site, and you can see that all of our samples cluster together pretty tightly, indicating that even over a distance about 30 meters on the ground, there's a lot of consistency in the soil chemical environment that plants have to deal with at that site. So this is in stark contrast to the nursery site, where chemically it's just kind of all over the place. And to really drive that point home, this plot, or this sample and this one, which are the most chemically dissimilar from one another in this analysis, are actually only 6 meters apart on the ground. So 6 meters is a pretty far distance for a caddy seed to move, because they tend to just kind of fall right underneath mom, but it's not very far at all for pollen to move. So this is the way I'm kind of thinking about these data so far, is that plants at the nursery site have to deal with strong selection. I mean, it's serpentine, right? Strong selection, but highly variable over pretty small spatial scales. Plants at the burn site may have to contend with stronger selection because higher heavy metals and lower essential nutrients, but at least that selection is fairly consistent over pretty large spatial distances. Okay, so back to our question about does gene flow increase or decrease fitness? So as a first pass at this question, we conducted a hand pollination experiment. This was last summer, so the end of that terrible drought. And at each, the burn and the nursery site, we had 40 randomly selected plants that we hand pollinated, either with pollen donated from another plant growing in that same population, 1 to 5 meters away from the recipient plant, so we call this local pollen, or foreign pollen, so burn plants receiving nursery pollen and vice versa. And then just to kind of verify these results, we had a second source of foreign pollen, which was plants from that Taylor population. Okay, so the expectation here is if there is strong local adaptation, plants should set more seed when they receive local pollen than when they receive foreign pollen. And if there is inbreeding depression, plants should set more seed when they receive foreign pollen than when they receive local pollen. Okay, so let's look first at the results from the burn population. When burn plants received local burn pollen, they set on average about 25 seeds per capsule. This is in contrast to when they received foreign pollen, and it didn't matter whether that foreign pollen came from either the Taylor or the nursery site, they set less than half as much seed. So this is consistent with a population with strong local adaptation or co-adapted gene complexes. To be honest, I was surprised to see this result so early on in terms of seed set, but there it is. And I want to be a little careful in making this suggestion, but it does possibly suggest that selection at the burn site is so strong as to have led to at least partial inhibition of admixture. Okay, so let's look at the nursery population now. So when nursery plants received local pollen, they set on average only about 10 seeds per capsule. When nursery plants received foreign pollen, again, it didn't matter if it was the burn or the Taylor population, they set about 2.5 times as much seed. So this is consistent with a population that is suffering from inbreeding depression, which is oftentimes defined very specifically as an increase in female reproductive success, and that's what seed set is. So this raises two rather obvious questions. First, is this population actually suffering from a loss of genetic diversity? And we're doing the molecular work right now to more directly measure that. And then why is this population losing genetic diversity? And I actually don't have any sort of good answer for that. But I do want to offer two thoughts on the matter. We have an ancillary data set, and I want to stress that this is an ancillary data set, but it suggests that there might be a higher rate of asexual reproduction at the nursery site than at other sites. So I mentioned that the plant can reproduce asexually by the splitting of bulbs, and these are 3 bulbs in various stages of splitting that were pulled from the soil at the nursery site. Again, it's ancillary data, but we saw an awful lot of this at the nursery site and we didn't see it at any other sites. This is destructive sampling though, and it's a threatened plant, so we're gonna have to go at this question with genetic techniques instead. Why this would be, I don't know, except that the nursery site really is kind of one of those classic severe serpentine balds, and the other sites aren't. And maybe there's something about the physical properties of the soil there that make perhaps establishment from seed less likely and may favor asexual reproduction. Another idea I want to offer is that it may not be that the population so much is losing genetic diversity, but equally as much because of the incredible variability in soil chemistry, the population may need to maintain higher levels of standing genetic diversity to cope with the amount of variability at this site. Okay, I just want to put these two results up next to one another because they're in striking contrast to each other, and yet these populations are only about half a kilometer apart. So these two populations are kind of doing very different things even though they're very close to each other on the ground. Okay, I want to now tie this to some of the work we've been doing on the population biology of this species. So we are 4 years into a 15-year-long demography study. And I'm going to show you lambda values from last year. Again, serious drought, and it was the year that this work was conducted. So the burn population was stable or grew by about 1.3% in that particular year. And the nursery population, though, decreased by 14%. Again, that's consistent with the population suffering from inbreeding depression. I really want to add that a lambda value is a snapshot in time. It's a snapshot of a much longer term process, particularly for a long-lived perennial like this. So it's not the whole picture, and I'm not saying that that nursery population is in rapid decline overall, but just during that terrible drought year, it did decline by about 14%. And LTREs indicated that that decline was attributable largely to a failure of seedlings to establish. Okay, so the real way, of course, to test for local adaptation is a reciprocal transplant experiment. This experiment just went into the ground in the past couple of, well, at the very start of the rains this year, so I don't have any data to show you yet, but we are working on this. But this will actually more directly test local adaptation, help us tease it apart from co-adapted gene complexes, and then whether or not genetic rescue, so the introduction of novel genetic material, might help this population we think might be suffering from inbreeding depression. We're also starting the molecular work to try and actually measure how much genetic diversity is in each one of these populations, degree of relatedness within and among populations, and then ultimately, hopefully, get to kind of spatial genetic structure for the species as a whole. Okay, I just want to end with a comment on the conservation implications of this work. And I think that Our findings here are broadly generalizable to most serpentine endemics, which account for an awful lot of the California natives of conservation concern. So artificial gene flow is kind of this hot idea in conservation biology right now, the idea of moving seeds, pollen, or sometimes even whole plants from one population to another, from a large stable one to a small declining one, to bolster it. And I would just urge an awful lot of caution with this particular approach. If there's strong local adaptation, we could be doing far more harm than good by doing this. On the other hand, if a population really is in decline because of a loss of genetic diversity, then this could essentially be genetic rescue, and it could be the difference between persistence or extinction of that population. But on that note, I want to emphasize that just because the nursery population is experiencing inbreeding depression does not mean that there's not local adaptation going on as well. And I just think that we need to be very mindful of the underlying genetic structure of these populations before we start moving genes around, because once you do that, you kind of can't get them back. Right? Okay, and with that, I want to acknowledge my funding sources, the Barrett Research Program, my team of research assistants, most of whom are here, one of whom was supported last year in part for her work on this due to a grant from CNPS. I think I've got time for questions.
So in terms of trying to limit genetic or moving genes around, how are you planning to treat your reciprocal plant transplants? Are you not going to flower?
So how are we avoiding doing that ourselves? It takes about 3 to 5 years for them to flower, so we've got plenty of time to measure things like germination, survival from year to year, before we have to start worrying about that. But I guess it depends a little bit on the results, but my inclination would be to remove flowers before they can spread genes.
And you're, in the nursery side, you had variable soils.
Yes, highly.
Is there any difference in seed set with the foreign pollen across that spectrum?
We have the data to answer that, and I can't answer it off the top of my head, but it's a really interesting and good question. Yeah.
Go ahead.
Okay. Yeah.
This may sound awfully old, but basically, are there any other species of Calicoris that could conceivably crossbreed with Tiburon?
I don't think so, and I think you'd have a hard time coming up for a justification of why that would be.
Well, because there's Calicoris luteus on part of the Tiburon.
There sure is. There's an awful lot of it. Yeah. As far as I know, that they are not cross-fertile, and I actually think that this population is pretty stable. I'm not worried. I also work with a streptanthus that I would not be surprised if it went extinct in my lifetime. I'm very worried, but I'm not worried about this particular species, so I'm not inclined to do anything radical like that.